not available, then on the basis of 3D QCR, quantitative structure activity relationship, and pharmacophore modeling, we can develop design new drug development. Structure-based, receptor-based drug design is generally these are the steps of the drug development. There's the disease identification first, drug target validation, then lead identification, lead optimization, preclinical trials, clinical trials, and drug approval, and its commercialization. This whole process takes more than 10 years if we go in detail. There are so many things. Use of medicine learning approaches by different steps in novel drug delivery system. Generally, everything is now practiced by the big pharmaceuticals and modern pharmaceuticals also. But it is a very difficult task to maintain the mm, other than the master chart, because the master chart is available in all pharmacies and it is the mandatory part of the GMP and Ayurveda ASU, Ayush department. So master chart you can find everywhere. But step by step monitoring is required for each and everything so that we can say that it is a standard. So array technology, proteomics, recombination technology, combinatorial chemistry, and novel techniques in the drug designing, and then pharmacogenomics. Now it is coming in the picture. There are so many technologies available nowadays, so we can focus on that particular area for the new drug development or for the repositioning of the drug. So, role of the pharmacogenomics at various stages of the drug development is very important. It aims to detect, monitor, and treat the molecular cause of the disease by using the various genomics technologies such as DNA barcoding, statistical genetics, and clinical development of the drugs. Many diseases developed as a result of the network of genes failing to perform correctly. Pharmacogenomics can identify the gene or loci which are involved in determining the responsiveness of the given drug. So drug target identification, phase one is the clinical trial, patient selection, inclusion, etc. You know everything. Many scholars have known, PG scholar and PhD scholars is known for this particular step, but it is a future because without knowing the pharmacogenetics and pharmacodynamics of the drug, it is very difficult to say something. So nowadays, it is a very common thing, very common question, how your drugs works. So generally, we explain it on the basis of our Rasaguna Veer Vipak, but it is a very difficult to explain on the basis of ADME study. So it's our approach should be in that way that they along with our traditional method we can focus on the modern side also. ADME study is required for the each and every formulation so that we can able to show the data, evidence based data. On the basis of that we can say that my drug is safe, there is no problem, you can move it. So so many things is required by the FDA also. Application of the proteomics is also very important nowadays. The proteomics is the complete evaluation of the function of the structure of the protein to understand an organism nature. Generally, when we are going to the chemical side or chemistry portion or biochemical side or biotechnology side, then we found this type of the activities going on everywhere. And many modern scientists are focusing on this particular area for the development of the new molecule, new drug combinations, new formulations. So, and it is a very effective one also. Certainly, so many things is coming in the market nowadays in the different forms, in the form of tablet, in the form of extract, in the form of gel capsules or nano suspensions, etc. are also coming into the market. Mass spectrometry is an essential tool that is used for profiling the proteins in the cell. It can identify the monitor biomarkers by analyzing the proteins in the body fluids such as urine, serum, exhaled breath, and spinal fluids. It can also facilitate the drug development by providing the comprehensive map of the protein interaction associated with the disease pathway. It is a common practice nowadays. We can use the saliva, we can use sweat, we can use urine or stool. Though it is also available in Ayurvedic literature also, that by seeing the characteristics of the saliva, urine, and mal, fecal material or sweat, we can assume what is the condition of Vata, Pitta and Kapha. On that basis, modern scientists nowadays searching for that. If we analyze the saliva, then we can focus on breast cancer, mouth cancer, other diseases are also indicative on the basis of the protein. On the basis of the protein. So all is good. Slide is moving now. So clinical proteomics.
Yes, sir. Slides are moving, sir. No problem.
Okay. Efficacy, resistance, and toxicity. Functional proteomics is multi-targeted drug discovery. Drug target discovery mechanism of drug action is also very important. So drug target interaction, specificity, and selectivity by the chemical proteomics. So many scientists are working on this. Generally, by using in IIT Jodhpur, we are also taking a note on this. In IUTech program, we are going for this chemical proteomics and functional proteomics. Clinical proteomics is in Delhi IIT, in ILVS in Delhi. Generally, we are focusing on taking the help of the other scientists and showing some extracurricular or extra activity of our drug molecules, and which molecule is effective and which molecule is not effective, in what condition we can say something. So these are the points which can be taken care, and for the future development also. Drug discovery, the natural products, chemical libraries, target discoveries, now it is available. Chemical libraries are available day by day. Number of chemicals are increases in the library, so that we can um, tabulate it and we found that the comprehensive result of that particular protein or gene or other metabolites. So metabolomics is a field dedicated to the high throughput identification and quantification of small molecules in biological system under the different conditions. Since the majority of the diseases are associated with the metabolic alterations, either as a cause or a result of the disease process, thus the identification of the change in the metabolic profiles can shed light on the mechanism of the disease and drug actions, as well as provide the useful diagnostic, prognostic, and risk factor of biomarkers. So on this basis, we can say so many things. Generally, systematic representation of different steps in the drug discovery and development process and examples of the metabolic implication in the process. Metabolomics can contribute during the drug discovery process, going from drug discovery and testing to response analysis, production, as well as treatment planning. So it is a very important nowadays, and many modern pharmaceuticals are focusing on this particular aspect, and they are going to develop from the natural to the synthesized product. Generally, natural products are available, but the quantity is the least and they are costly. Then it should be converted into synthesized or artificially prepared in the laboratory. So there are so many things coming in practice nowadays. So many nutraceuticals are coming with the extract form or with the uh, solution form in the market on the basis of this only. So it is also a very good technique to use in the drug discovery system. Network pharmacology and emerging technique for natural product development is a scientific research on Ayurveda, because generally network pharmacology, because initially we have gone through this book to this book, this book to this, different references should be collected physically, but nowadays it is available on the Google in network pharmacology. Just to point out this some herbal drugs or herbal molecule, then you can find so many things on your screen. So everything is available. Which one is synergistic, which one is the antidote. So you can also found in that particular manner. So network pharmacology is very important nowadays. So because whole plant is not required for just we require some solution, and GCMS, by this GCMS we can Banyan chemical database match this chemical structure, and we found which type of the compound and which type of activity is seen in this particular form. So component target protein network is available, protein-protein interaction and geo-analysis and KEGG analysis is also important. Target protein pathway network. At last, we can found the target protein pathway. If we can found the target protein pathway, then we can check it, the pathology, or treat it in such a manner that it can be reversed. So on this basis, so many drugs are coming nowadays in market: anti-diabetes, anti-inflammatory, ADME scanning, potential compounds, database screening, searching, and potential target proteins. Because we know that the mechanism, the biochemical, our metabolic process is totally dependent upon the different proteins, different test elements. So if we can assess it in the form of the protein or metabolites, then we can search a molecule which is more effective than our traditional one, natural one. So, but everything is there. But as far as our knowledge comes, then we saw that if the activity of the any compound is increases in manifold, then the toxicity or adverse effect also increases in the same way. So very cautiously it can be implemented in our science. Otherwise, it creates problem for us also. So this is a picture of the Triphala network pharmacology. You can find individually materials are in different structure. But when it is combined, all three combined, then the structure of this Triphala is different. And something new is developed. Something is new developed. So this new thing is more efficacious for us. So synergistic action is everywhere accepted, and due to this, the activity of the each molecule or each plant is synergized in such a direction that is we require. So it is a very good module for the new drug development. Phytogonous phytoconstituents of the Bidania somnifera annulled the asugandhanolide as per the potential drug targeting breast cancer. Investigation through the computational molecular docking and conceptual DFT studies. अश्वगंधा को हम कभी सोच सकते थे कि वो ब्रेस्ट कैंसर में काम करेगी? नहीं, लेकिन जब मॉलिक्यूलर डॉकिंग हुई तो उसमें ये नई स्टडीज हमको मिल गई। तो ब्रेस्ट कैंसर में भी इस तरीके से इस तरह के स्टडीज जब मॉलिक्यूलर डॉकिंग की बात हम करते हैं तो हमको मिलती है। सो वी कैन मूव ऑन द बेसिस ऑफ दिस कंपाउंड डज नॉट पॉसेस एनी mutagenicity, hepatotoxicity, and skin sensitization. Based on the result obtained, the present study highlighting the anti-cancer potential of the ashwagandha, a compound from Begonia somnifera. Furthermore, in vitro and in vivo studies are necessary to perform before clinical tests to prove the potential of the ashwagandha analyte. So it is a must. Directly we cannot go for the human being. So in vivo and in vitro study is required, but certainly we found if we calculate it, then certainly we minimum reduces that 5 to 6 years. So for the drug development, it is this type of the studies is required. So immunoprotective potential of the Ayurveda have calmate by LCMS and MS and network pharmacology analysis. This paper is published, so you can please go through it. The GCMS of the network pharmacology analysis is Ayurvedic fermented medicines Chandanaso against the chronic kidney and cardiovascular disease. It is also published, so please go through. Drug repurposing and emerging approach for the novel diseases. जनरली हम ड्रग देते हैं किसी काम के लिए, पता चलता उसमें तो कोई फायदा नहीं मिला, लेकिन कोई दूसरे सिम्टम्स सबसाइड कर गए। अब वो ड्रग उसके लिए बताई नहीं गई है, लेकिन हमने देखा कि उसमें वो इफेक्ट मिल गया, तो जनरली अपने प्रैक्टिस के आधार पे हमने वो ड्रग यूज करनी शुरू कर दी, और होता है कि उसके रिजल्ट बहुत अच्छे मिलने लगे और वो प्रैक्टिस में आ रही। तो दिस काज एज ए कॉल्ड एज ए ड्रग रिपर्पसिंग। और उसमें जनरली मैक्सिमम चाहे जो नेटवर्क फार्माकोलॉजी है, जो हमारे प्रोटोमिक्स है, जो मेटाबोलोमिक्स है, ड्यूरिंग दिस स्टडी वी फाउंड so many drugs are under the repurpose. We have searched so many activities, but main activity is aside, but there are so many activities also comes in the form of the outcome. So a process of identification of new pharmacological identification pharmacological indication from old oblique existing or failed, many time failed investigational or already marketed or FDA approved drugs, products and application of the newly developed drugs to the treatment of the disease other than the drug original intended or therapeutic use. Also known as a drug repositioning or drug retasking or drug recursing or drug recycling or drug redirection, therapeutic switching. These are the terminologies given by the different scientists for the same. So advantage of over the traditional drug discovery is significant reduction of the time spent in the R&D. Cost effective, reduce risk failure rate is more than 45% is associated due to safety and toxicity issues in the traditional drugs. Highly efficient strategy, maximizing the therapeutic value of the drug. So many things is going on everywhere. So it should be adopted in our mind also, that anything can be done by using the different technologies. Now it is technologies available, and younger students or younger scholars or younger scientists are coming with a different mind. Generally, they are not prejudiced, so they incorporated many things in their mind, and from that we can find so many things. So this is the example, repurposing of the potential of Ayurvedic medicinal plants derived active principle against the SARS-CoV-2 वायरस के लिए दिस इज यूज्ड। कोविड में वायरस यूज किया था और पता चला, लेकिन हमको मटेरियल आउटकम मिला, वो जाके कहीं और मिल गया जाके। So immunity, molecular docking, molecular dynamics, and MMPBSA study shows that it is a viral protein required for the replication with a nonon is also predicted to be the block the cell membrane receptor required for the entry of the virus to the cell. अगर हमको ये मिल गया तो हम इसके आधार पे बहुत सारी चीजें अपनी रोक सकते हैं। तो हमको एक वे ऑफ टैकलिंग मिला इसमें कि कैसे काम कर रही है, किस प्रोटीन के थ्रू काम कर रही है। अगर ये मिल जाता है तो उस प्रोटीन को हम ब्लॉक कर सकते हैं, उस प्रोटीन को वहां से कम कर सकते हैं, ज्यादा कर सकते हैं, जिसके आधार पे हम मैक्सिमम रिजल्ट्स मिल सकते हैं। सो डेवलपमेंट ऑफ न्यू डोजेस फॉर्म्स, नैनो मेडिसिन, नैनो emulsions, नैनो सस्पेंशन, नैनो मिसलेनियस must be the following the characteristics: reducing the toxicity and minimizing the side effect of the drug by targeting them to a safety site of the action, reducing dose through the improved bioavailability, reducing dosing frequency by controlling the drug release into the human body, improving the shelf life by enhancing their stability, increase patient compliance. So these are the benefits of the nanomedicines. Nanoformulations used to improve the effectiveness of the natural compounds. It is also proved nowadays because there are so many things on the quality control and shelf life. And these nanomedicines are quite safe and quite long shelf life. So enhance, improve the stability of the controlled release, enhance the reserve memory and learning impairment, increase the uptake and concentration in the brain, improve the pharmacokinetics, increase the bioavailability, enhancement of the neuroprotective activity. Everything is given in the slide. Herbal nanomedicine's recent advancement, challenges, opportunities, and regulatory overview. This paper is published by the Mr. Teja et al. In the review, authors concluded a combination of the herbal medicine with the nanotechnology can be essential tool for the advancement of the herbal medicine research with enhanced bioavailability and fewer toxicities. Despite the challenges related to the traditional medicine's safe and effective use, there is a huge scope of the nanotechnology based herbal medicines. Overall, it is well established that herbal nanomedicines are safer, have higher viability, and have enhanced therapeutic value than conventional herbal and synthetic drugs. It is well established, but when go for the nano herbals, then so many things is also going on, every parallel by parallel. So this is the nano formulation of herbal extract in the treatment of the neurodegenerative disorders, and this found some things in nano form. Self-nano-emulsifying drug delivery system for the embelline design characterization and in vitro studies. However, the viability of the embelline is extremely erratic due to the inherent poor aqueous solubility and dissolution properties. With the objective of improving the poor solubility and dissolution properties of the drug, advanced drug delivery system are requisite. कोई भी नई चीज होती है तो उसके लिए भी हमें बहुत सारी मेहनत करनी पड़ती है, ऐसा नहीं होता है कि एकदम से हो जाती है. सो इट इज द आउटकम ऑफ द नैनो इमल्सिफाइंग capsule snack 30 curcumin emulsion is available here, and it is very popular nowadays for the allergic disorders and for the immunity. So many persons are using this. It is in the form of the capsule. So development of safe drug through the novel approaches of the toxicological screening is required. Mutagenicity testing to assess the submicroscopic changes in the base of the sequence of the DNA, chromosomal aberration and structural aberration in DNA, including duplication, insertion and inversion of and translocation. Development of the developmental toxicity or embryotoxicity studies, neurotoxicity, one generation to reproduction toxicity testing, toxicokinetics to study the metabolism and execution of the pattern of the higher doses of the drug. So it is must nowadays. Otherwise, we found something very absurd one. So neurotoxicity and mercury chloride, methyl mercury and cinnabar containing this and this in the rat comparative studies. So many papers are published. Outcome of these papers are when these formulations were screened for their safety studies, they are found to be less toxic or not toxic as compared to other environmental mercury and arsenic toxicants. The tissues accumulation of Hg and arsenic is found to be very minimum to the cause of the toxicity and different biomarkers in nephrotoxicity. So, on the basis of that, it is an interesting fact that half of all drug candidates fail in the late stage of the clinical trial. So many persons coming with the R&D facilities and R&D new drug developments, but 50% of the drug fails in the clinical trials. And it is recorded everywhere. If you go for the CDRI, TC, so very big institutes, then you find the same results. And soon after their approval, some of them have to be withdrawn from the market due to their severe side effects and clinical risks that are not detected in the phase 3 third trial as per the pharmacovigilance studies. The average cost to reach the develop reach the average cost of to research and develop each successful drug is essentially estimated to be around $800 to the $1 billion. So it is very high. Everyone cannot afford it. Given the development of the synthetic chemicals for a therapeutic use is by and large a random process that might result in screen serendipitous discovery. Many pharmaceutical companies are now focusing on the development of the plant-derived products. You can see from this picture, if 250 compounds we have screened in the drug discovery and preclinical trials, after 5 years and 6.5 years, they have gone to for the clinical trials. Then only 5 compounds goes in the phase third, and after 6 years, 6 and 12 years, 12.5 years, then only once is finalized and given approval from the FDA. It will again takes more than 4 years. So around 16 to 17 years for the development of a new molecule. So it is a very difficult task for all of us. So it is a very good thing for us that all classical literatures are excluded from this. We can think on that particular aspect, and we found some very good explanation on the basis of the observational studies which had been done by our ancestors and given in such a form, punch with in the form of kasaya or in the form of formulations or in the form of single use of the drugs that can be used by us in different conditions, then we can find a very good results for everything. So today we have given some exposure for the new advancements which has been accepted by the modern scientists, modern pharmaceutical companies, and some of our Ayurvedic manufacturing companies are also in the process. They are adopting same things in their R&D facilities. So on that basis, probably we find that our knowledge, our tradition is quite good, and everything is written in our literature. But again, after the analysis of the quality, after the analysis of genuineness, after the analysis of their uh, therapeutic and efficacy and safety, we can use everything in such a way that drug should be used in prescribed conditions, in prescribed doses with Anupan and Sandpan. Then it is 100%, we found 100% success in our clinical trial without any adverse effect. If any adverse effect is found, then please report to the pharmacovigilance center so that we can move forward for further action. So thank you to all of you for patience and um, calm and cool in learning, and thank you to the Multani Pharmaceutical for giving the platform to say something, especially Pradeep Multani sir, because he is also my senior. So I respect all the time for his views, and his generally we follow the order. So thank you, sir.
So, absolutely, simply outstanding, sir. Thank you very much. I'm sure all our people who have joined have really learnt, and so have I. So, very, very obliged. Thank you, sir. Dr. Minakshi, over to you.
Yes, sir. Thank you, sir. Thank you, sir. Thank you, Prajapati, sir, for your elaborate lecture on emerging novel techniques in the field of herbo drug development. आपने हमें बताया सर कि role of genomics at various stages of drug development is very much essential, and it should be done with pharmacies to develop new formulations or to work on the ancient formulations. It should be done. Then application of proteomics, metabolomics, GCMS network pharmacology is also mandatory nowadays. Matlab it should be done, and it should be cautiously used, and cautiously it should be included in Ayurveda drug discovery. थैंक यू सो मच सर, क्योंकि ये सब टर्मिनोलॉजीज के ऊपर हमने काम करना है, और एक अच्छे आयुर्वेदिक मेडिसिन मार्केट में आए, इसका ध्यान रखें। तो आपने ये बहुत अच्छे से इसका हमको ज्ञान दिया सर, और आपने बहुत ही राइटली कहा सर कि बिग सक्सेस डज नॉट कम विदाउट बिग चैलेंजेस। सो एंड हाउ वी डील विद द चैलेंजेस मेक्स ऑल डिफरेंसेस। Yes, sir. Very rightly said. And thank you so much. Your knowledge and experience will definitely guide the scholars and learners of Ayurveda. So, once again, I request all attendees and scholars of this webinar kindly drop their names and email IDs in the chat box so that we can mail your e-certificate without missing anyone. And now the webinar is open for the question answer session. Respected scholars, students, and respected audience may unmute and ask their queries, or you may post them in the chat box also. आप अपने क्वेश्चंस पूछ सकते हैं अपना माइक अनम्यूट करके। मैं सभी अटेंडीज को रिक्वेस्ट करती हूँ कि आप अपने क्वेश्चंस पूछ सकते हैं। Now the session is open for question answer. You may drop your question.
डॉक्टर, अभी हम देख रहे थे कि इसमें बहुत सारे नाम, बहुत पुराने स्कॉलर्स भी हमारे जुड़े हुए हैं।
जी जी जी
अब
जी,
देख रहे हैं
श्रद्धा मैडम भी आए हैं।
यस, so many persons are there. If anyone says something, then it is the most welcome. आप इसमें बोल भी सकते हैं, कह भी सकते हैं, अपने सजेशंस भी दे सकते हैं, क्योंकि आपके भी एक्सपीरियंसेस अभी बहुत कुछ हो चुके हैं। हम देख रहे हैं श्रद्धा भी जुड़ी है, तिवारी जी भी, मानसू भी जुड़े हुए हैं, भाग्य लक्ष्मी भी जुड़ी हुई हैं, और सीनियर पर्सन्स हैं जो लोग जुड़े हुए हैं। तो
I request all senior persons kindly share their experience and a few expressions kindly. Your expressions are welcome. Please express yourself, ma'am and sir.
दोबारा निकाल रहा हूँ।
I request all
Good evening, everybody. Am I audible?
Yes, very much audible, ma'am.
The sole intention of I being joining this webinar today was to have a darshan of my respected guide sir, Professor P.K. Prajapati sir, and just wanted to hear from him after so many years. And I was really thankful the way he explained and shared his expertise.
Yes, thank you, ma'am. आप जितने स्कॉलर्स और लर्नेड पर्सन जुड़े हैं, और वो इनकी जो भी क्वेरी है, आप अपने क्वेश्चंस पूछ सकते हैं।
Ma'am, should I?
Yes, yes, please, ma'am.
Thank you.
Yes,
very
yes, ma'am.
Yes. Sir, very knowledgeable sir it was. Sir,
Ma'am, you are not audible clearly, please.
Hello.
हाँ, now
Is
you
it
are audible,
clear,
ma'am.
sir?
Yes, yes.
Okay. Sir, I want to get some knowledge regarding nutrigenomics, sir. Not focused on genomics, so omics. So nutrigenomics, if we can work with nutraceuticals product.
हेलो प्रजापति सर,
सब
अनीता मंजारी मैम वर्धा से ज्वाइन हुए हैं सर।
हाँ हाँ, यस यस, क्वेश्चन क्लियर नहीं हो पाया, लेकिन ये है कि हमें लगता है कि वो ओमिक टेक्नोलॉजी पे,
न्यूट्रोजेनोमिक्स के बारे में जानना चाह
हाँ,
रहे हैं सर।
न्यूट्रोजेनोमिक्स के बारे में जानना चाह रही हैं।
जी।
ऑलरेडी देयर इज वर्क इज गोइंग ऑन इन एफ ये दिस इज NIN Hyderabad, yes. NIN Hyderabad,
Okay.
the work is going on there. If you have some contact, then otherwise you drop a mail, then they have
Yeah.
give you the reply on this particular issue. अनीता जी, वो होता है क्या है मैक्सिमम टाइम में कि जो अभी हमारे पास लाइब्रेरी है ना, इसमें जेनोमिक्स में
जी।
ये इम्पोर्टेंट हो जाती है कि हमारे पास लाइब्रेरी कैसी है। जितने भी लाइब्रेरी अवेलेबल होगी तो उसके रिजल्ट तो मिल जाएंगे। अगर उसकी लाइब्रेरी अवेलेबल नहीं है तो फिर रिजल्ट मिलना मुश्किल है। सो इन फूड प्रोडक्ट्स एंड न्यूट्रिशन में जो उनको काम दिया गया है वो हैदराबाद को ही दिया हुआ है, तो हैदराबाद इज डूइंग समथिंग, तो वहां से आप इसमें ले सकती हैं उनसे। हम्म।
जी सर, थैंक यू वेरी मच सर। सर मैं वर्धा
थैंक
से
यू।
हूँ, और आप महात्मा गांधी आयुर्वेद कॉलेज वर्धा में आए थे सर पहले, मतलब काफी साल पहले।
हमें याद आ गया।
जी जी जी।
यस यस
सर
मैडम।
नमस्कार, चरण
नमस्ते
स्पर्श।
नमस्ते नमस्ते।
डॉक्टर अनीता मंजारी मैम मेरे गाइड और HOD है सर डिपार्टमेंट के, जब मैं
अच्छा।
MD कर रही थी।
नहीं, अभी
यस।
एचओडी नहीं है सर, मैं बट इनकी गाइड जरूर रही हूँ। डॉक्टर रोहित एंड डॉक्टर प्रशांत they were along with me long before.
यस यस, धीरज एंड रोहित।
Yeah.
यस। मैक्सिमम टाइम होता ये है कि ना हम लोग बहुत बड़ी बड़ी बातें तो करते हैं, बहुत बड़े बड़े चीजें भी देखते हैं, लेकिन हमें लगता है कि हमने आप लोग को अपडेट रहना चाहिए। लेकिन अभी
जी
तक के
सर,
जो
जी
भी
सर।
हमने थिंग्स पाई हैं उसमें हमें जितने भी अपने ट्रेडिशनल वे ऑफ प्रिपरेशंस है ना, वो ज़्यादा सूटेबल
हम्म,
मिले हैं देन कि हम जो अल्टर कर रहे हैं। अभी थोड़े दिन के बाद उस पर भी कोई ना कोई स्टडीज हम लोग करेंगे, क्योंकि अल्ट्रेशन एडिशन में कई चीज़ें निकल जा रही हैं, गायब हो जा रही हैं, और वो जिस फॉर्म में हमें चाहिए उस फॉर्म में मिलती नहीं है। हमने यही बोला था कि जितनी जितनी हम टॉक्सिक एक्टिविटी बढ़ाते जाते हैं किसी भी प्रोडक्ट की,
हम्म,
साइमल्टेनियसली उसका जो एडवर्स इफेक्ट या उसका साइड इफेक्ट भी, क्योंकि हमने एक तरफ पोटेंसी वन डायरेक्शन की है, तो वन डायरेक्शन में पोटेंसी जब करेंगे तो दूसरे डायरेक्शन में उसकी प्रॉब्लम भी बढ़ेगी, क्योंकि बिल्कुल
जी
टारगेटेड आर्गन पे जाके काम करेगा, और टारगेटेड आर्गन पे करेगा तो जो हमारा कंसेप्ट ऑफ होलिस्टिक अप्रोच वाला था वो मिस हो जाता है उसमें।
जी जी जी।
तो हम
यस यस।
कई बार ऐसा पाते हैं कि एक प्लांट्स के अंदर अगर हमारे पास एक्टिविटी है तो उसको एक लेवल तक रोकने के बाद, एक लेवल तक बढ़ने के बाद उसको रोकने के मॉलिक्यूल्स भी वहीं अवेलेबल होते हैं, और वो उसको
यस
रोक देते हैं जिससे कि टॉक्सिटी नहीं होती दूसरे ऑर्गन में। लेकिन ये क्या है कि अब जमाना अलग है। पहले हम लोग बाजरे की रोटी और चने के साग खा लिया करते थे, मक्के का साग खा लेते थे। आज बच्चों को तो पसंद नहीं आते, क्योंकि उसको चबाना ज्यादा पड़ता है, मेहनत ज्यादा करनी पड़ती है। अभी तो कैप्सूल का जमाना है कि हमारा कुछ भी सप्लीमेंट है तो आप कैप्सूल लीजिए, अंदर गया और उसने दे दिया, तो कितने दिन तक हम उस पर डिपेंडेंट रहेंगे। लेटर ऑन हमारी आदत हैबिट में बन जाएगा, और हैबिट बन जाएगा तो जो हमारा इंटेस्टाइन का है काम वो नहीं होगा, फिर हम ILBS से शिकार होंगे। तो ये कई चीजें
यस
हैं जहां हमें इन चीजों को देखने की जरूरत है कि हमारे नेचुरल हैबिटेट में बहुत सी चीजें, लाइफ स्टाइल डिसऑर्डर की हम बात करते हैं तो रीजन और सीजन के हिसाब से डाइट एंड लाइफ स्टाइल को मेंटेन करने की बहुत जरूरत है। ये मेरा पहला व्यू रहता है, क्योंकि अगर हम उसको मेंटेन करते हैं तो बहुत सारी चीजों को हम कम बैक कर जाते हैं तुरंत। तो रीजन और सीजन के हिसाब से अपने लाइफ स्टाइल, डाइट स्टाइल को मेंटेन करने की कोशिश करें। और अपने यहाँ कहा भी गया है औसत तो बाद में आती है, परम औसत भोजन ही है हमारा, पथ्य अपथ्य जो भी है, तो अगर उस पर हम फोकस करें तो और अच्छी बात है। वहाँ भी बहुत सारी चीजें आ रही हैं, बहुत डेवलपमेंट हो रहे हैं। इस प्लेटफॉर्म से थोड़ा सा हमें भी बोल देते हैं, क्योंकि जनरली हम कई बार बोलते हैं कि 1 बीघा के अंदर, 1 हेक्टेयर के अंदर कुछ पहले 10 क्विंटल गेहूं हुआ करते थे, अब आज उसी 1 हेक्टेयर में 100 क्विंटल गेहूं हो रहे हैं। जमीन तो वही है, हमने ऊपर से मटेरियल दिया, ऊपर से डाला, कितनी क्वालिटी हम मेंटेन कर पाएंगे। क्वांटिटी बढ़ेगी तो क्वालिटी मेंटेन नहीं हो सकती, दिस इज द फैक्ट। आप एवरीवेयर कहीं भी हम देख लेते हैं कि जहाँ जहाँ क्वांटिटी बढ़ती है वहाँ क्वालिटी मेंटेन नहीं हो पाती। तो क्वालिटी मेंटेन करने के लिए हमको क्वांटिटी और क्वालिटी दोनों पे ध्यान देना पड़ेगा, अदरवाइज प्रॉब्लम हो गई। और इस समय ये हमने ऑर्गेनिक का एग्जांपल दिया आपको कि ऑर्गेनिक टमाटर टमाटर मार्केट में 60 रुपए किलो है और ऑर्गेनिक टमाटर आपको 100 से नीचे नहीं मिलता है, लेकिन हम ऑर्गेनिक के नाम पे ऑर्गेनिक खरीद रहे हैं और ऑर्गेनिक है कि नहीं है हमें कुछ पता नहीं है। तो काफी चीजें हैं, ये मार्केट है, मार्केट में सभी चीजें की अवेलेबल है, लेकिन हमारा सिर्फ इतना कहना है कि हमको सबको एसेस करके प्रैक्टिकल उनके नॉलेज को गेन करके चीजें जो सुटेबल है उनको यूज करना चाहिए, जो सुटेबल नहीं है उनके बारे में सोचना चाहिए कि क्यों नहीं है। उनको बाहर नहीं फेंकना है, सोचना है और उनको भी एक्सेप्टेबल फॉर्म में लेके आना है। दिस इज आवर ड्यूटी।
वेरी वेल सेड सर। सचमुच हमको आज के मॉडर्न टेक्नोलॉजी के साथ में एंशियंट विजडम का साथ हम हमेशा ही साथ में रखना चाहिए, ताकि हम डिसाइड कर सके कि हम किधर जा रहे हैं करके, और एक्चुअली हमारी हेल्थ के लिए क्या बेनिफिशियरी है करके। तो एंशियंट विजडम कभी भी फीका नहीं पड़ सकता है सर। और आपने सही कहा कि हमें दोनों तरफ ध्यान रखना है। आज जो नए बच्चे हैं उन्हें न्यू टेक्नोलॉजीज ज्यादा समझ में आती है, लेकिन वो जब ये बच्चे जब ज्ञानी होकर चीजों को समझकर जब एंशियंट विजडम की भी सारी चीजें समझेंगे तो वो बेटर डिसाइड कर सकेंगे कि कौन सी चीज बेस्ट है इन दोनों को मर्ज करके। तो न्यू टेक्नोलॉजी की तो हमेशा जरूरत रहेगी, और इसी से बहुत अच्छी प्रोग्रेस भी होगी सर। थैंक यू सो मच। आई थिंक और किसी को अपने थॉट शेयर करना है तो मैं सर वेलकम करती हूँ। प्लीज आइए और आप बता सकते हैं या अपने शेयर कर सकते हैं। I think no more queries from now the audience, sir. So we may proceed further to the next segment, which is of acknowledgement. Before that, I once again request dear audience to kindly put your names and email ID in the chat box so that we can mail your participation certificate. Expression of gratitude is an integral part of any organized program. I invite our research officer, Ms. Jyoti Singh, to deliver vote of thanks for today's webinar. Thank you. Please, Jyoti.
Thank you, ma'am. Good evening, everyone. I feel very privileged to be here on this encouraging occasion and to be asked to deliver a vote of thanks on behalf of our Multani Pharmaceutical Limited at today's webinar, Emerging Novel Techniques in the Field of Herbo Mineral Drug Development. On behalf of Multani Pharmaceutical Limited, I would like to express my gratitude to Honorable Chief Guest, Professor P.K. Prajapati sir, Honorable our respected Chairman, Dr. Pradeep Multani sir, and all esteemed delegates of the webinar for their presence and contribution to make this webinar a great success. As we all know that saying thank you is a respectful way to show appreciation towards a person, but these words would be less for today's chief guest and speaker, Professor P.K. Prajapati sir, who is Vice Chancellor, Dr. Sarvapalli Radhakrishnan, Ayurveda University, Jodhpur, Rajasthan. His talk not only gave us knowledge about Ayurveda science, but also taught how to manage the difficult times with Ayurveda. A true teacher uses their knowledge in the development of the nation, which could be clearly understood from his lecture. Next, I would like to thank Dr. Pradeep Multani sir, Chairman Multani Pharmaceutical Limited, for providing immense support to make this webinar successful. Thank you, sir, for inspiring and encouraging us with your words on this occasion. Learning is always a good opportunity to feed our minds as it helps us grow. Through such informative seminar, we get chance to boost our knowledge with the new things. So I would like to take my time in thanking our dear audience for their active participation. I must thank the organizing team, volunteer for working hard for the last few days to make this webinar successful. Thank you everyone once again, making it great success. Thank you so much.
Thank you, Jyoti. Now,
No,
with
thank you
Chairman's
very much.
permission. Ji,
Yeah,
sir.
please carry on.
Ji, sir.
Anji, Dr. Meenakshi.
Ji, sir. Thank you, sir. Now, with Chairman Sir permission, I declare that today's webinar is complete, and we will meet again. Soon after in our next webinar session. Thank you. Jai Hind, Jai Harry. Thank you, sir.